buddies, you know. But Dr. Gold is the director of the University of Florida Center of Translational Research in Neurodegenerative Disease, as well as a professor of neuroscience in their College of Medicine. He's exciting, and I think you're going to be excited to listen to his story.
Well, thank you, Nick, and you know, it's been a great pleasure to come up and see your wonderful facility, and I know that, you know, I think it, given the history of what's happened with your father, and it's a great memory, and I know he would be proud to see it today. And what I'm going to try to do today is to try to, you know, Dave set the problem up. This is our epidemic. This is the challenge that the baby boomers are facing. But why is there hope? What is our hope? And so what I'd like to do is tell a story. And this is the story, it's a murder mystery story. And it's the murder mystery story about Alzheimer's disease. And it began really 105 years ago with a German physician named Dr. Alzheimer's. And he had his first patient was a woman named Auguste D., who presented with a feature of reduced comprehension, so that's the dementia, memory, but she also had a number of other symptoms, paranoia, you know, unpredictable behaviors. And for anybody who suffered or watched a family member go through this, it's not just the memory loss, it's not just the cognitive impairment, it's also these other symptoms that make Alzheimer's such a devastating disease. Not only do you watch somebody lose their humanity to a large degree, but you also have unpredictable behaviors that are tough to control, and it's really hard to manage. And so that was the initial index case, and that's where the name Alzheimer's disease came from. But many of here, I'm sure now, it's easier to talk about this with CSI and other things on TV where you have actually pathologists. I'm trained as a pathologist. I did my PhD in a pathology department and my medical training in a pathology department. Nobody knew what was the cause of Alzheimer's disease, and Dr. Alzheimer's did a brain autopsy on Auguste D. And at that time, he described two major lesions. Not only was the brain shrunken and atrophied, so I call it brain organ failure, but there were also two lesions called plaques and tangles. And the plaques and tangles were the first description that Dr. Alzheimer's made. So what happened between 1906 and the mid-1980s? Really not a lot. And so there was a big gap in our knowledge. So we knew that somebody was killing people named Alzheimer's disease, or what we call Alzheimer's disease, but we didn't really know how to place that in the context of other forms of dementia, or whether these lesions that he saw had anything to do with the disease. So what happened? Well, in the 1980s, people actually figured out what the proteins were that were in these lesions, the plaques and tangles. And they turned out to be two different proteins. One was called amyloid, as Dave referred to, and the other was called tangles, or tau, that's in tangles. So we had these two proteins, and we knew that they were in these lesions, but that didn't prove cause and effect. Could they be just markers or graveyards or tombstones? And so this is when Dave and I really came into the field. We came into the field where now we knew what these proteins were, and there were some people who argued, well, this caused disease, and some people caused this caused disease, but nobody really knew. So what helped us? What was, just like in many other diseases, what helped us was genetics. It turned out that there were rare families, and many people don't know this, but there are rare families in the world where Alzheimer's disease is what we call dominantly inherited. That means 50% of the children get the disease. So, and it typically in these genetic forms of Alzheimer's disease, people get it younger. So they don't get it when they're 65 or 70 or 75. They get it when they're 40 or 35. And so you can imagine how devastating this was. But we were able to figure out, or scientists in the field were able to identify the genes that caused the Alzheimer's disease in those families. And guess what? It pointed us right back to amyloid as the culprit. So in many cases, there are mutations in the gene that makes the amyloid protein that can be shown to cause Alzheimer's disease. In other families, there are different genes that were mutated. But again, impacted whether this amyloid protein would deposit in your brain or not. So this was really the roadmap where we went from, well, this is a disease, we don't know what's causing it, but we know that this is what happens to your brain at the end, to saying, no, wait, this is really the trigger. So what I like to say is amyloid is very much, as David alluded to, like the cholesterol of Alzheimer's disease. Except it's a protein we normally make, it deposits in your brain, and it triggers a complex cascade of events that ultimately lead to dementia and brain organ failure. Now, what about tangles? Dr. Alzheimer's also described these lesions. Well, it turns out there are people who get another form of dementia called tangle-only dementia or frontal temporal dementia. And in those families, mutations in this gene that make the tau protein actually cause the disease. So it tells us, I'm sorry, if anybody was asleep, they're not anymore, that the tau protein is also important. And in fact, in the scheme of events that we now know to occur in Alzheimer's disease, you get amyloid deposits long before you show symptoms. Subsequently, tau begins to accumulate in these tangles. And then later on, your brain begins to atrophy and you begin to show symptoms of the disease. And we think this continuum takes 15 to 20 years. So what does that help us with? The other thing that's important to know, and this question comes up many times, is, well, what's the difference between dementia and Alzheimer's disease? And this is an important distinction. And by understanding these different forms of dementia, what we now can do is subclassify them. And we could say about 60% of all dementia, so people who suffer cognitive impairment, it's a clinical term. It's not a diagnostic term in terms of what kind of dementia do you have. We could say 60% of those people have Alzheimer's disease. And now with the use of scans and even measures where we could actually take a little bit of fluid from your spinal fluid and measure certain proteins, actually the amyloid and tau proteins in your spinal fluid, we could be pretty good about telling you that you have Alzheimer's disease as opposed to other forms of dementia. So what are these other forms of dementia? Well, a lot of it is vascular. And vascular dementia encompasses a whole bunch of different types of terms, but essentially it means that you've had a lot of small strokes that sort of accumulate in a dementia. And then there are a whole host of other diseases, such as frontal temporal lobar dementia with Parkinsonism, and complicated names and worse names than that compromise a very small portion of other forms of disease. But yet, these diseases, even though they're rare, are no less devastating. And about 10% of Alzheimer's disease is traditionally misdiagnosed, and in fact, somebody is suffering from one of these other forms of those diseases. So, how does that help us? Well, we had a roadmap. We know what causes this disease, and we know what it leads to. So in truth, what it's told us is how to prevent the disease. Much like the linkage, so people don't really know that how did we ever figure out what happened with cholesterol and heart disease? Well, it really started in the Korean War, when some young men were killed on the battlefield, and autopsies were done in the Korean War, and they looked at their major vessel, the aorta, coming out of the heart. And inside that, they found that there were these fatty streaks. There were these fatty streaks, and in those was cholesterol. And so some people were arguing, well, this couldn't have anything to do with heart disease because it occurs in these young people, and they're not going to have a heart attack for 20 or 30 years or 40 years. But again, genetics came to the rescue. It turned out that several people won Nobel Prizes for figuring out that there were families who had high cholesterol levels, and there were genes that were mutated in those families that caused them to have those levels, and they would die of heart attacks in their 30s or 40s. And that enabled us to develop, or the medical field, to develop drugs that lowered cholesterol and provided the rationale for testing those in humans. And we now all know that those are highly successful drugs. They don't completely prevent heart disease, but they have a big impact, especially if you start early. So this roadmap in Alzheimer's disease, where we now know amyloid is the trigger of this disease, means that if we can lower amyloid or prevent it from depositing in the first place, then we can prevent this disease from happening. And the challenge, though, is that in the regulatory environment that we deal with today, where we could say, okay, if I had a drug today and I could show it lowers amyloid in somebody 20 years before they had symptoms of the disease, would it get approved? And the answer is probably